The 21st century brought a reawakening of Ferrier's methods and results, with Graziano's ethological action maps for the motor cortex. In 2002, Michael Graziano of Princeton University had one of his colleagues burst into his office quite animated, saying, "You've got to come and see this." Graziano and his colleague were studying a brain region called the frontal eye field, which is involved in controlling eye and head movements. His colleague had inserted an electrode into the brain of one of their monkey subjects to investigate the frontal eye field, but had made an error in location, and electrical stimulation to this region evoked a motor response of the monkey's arm. Subsequently, Graziano and team followed this minor error to fully study microstimulation of the motor cortex, because what they had discovered were complex arm movements, a return to Ferrier results of some 130 years earlier. Under electrical stimulation, the monkey's hand had moved to its mouth, and more than that, the index finger flexed onto the thumb, the forearm supinated, and the mouth opened. Both scientists were stunned, and this little mistake spawned a major breakthrough in the understanding of the motor cortex. Depending on where they inserted their electrode, they produced a range of functional action patterns for the monkey, like defense, reaching, and chewing. Ethological means that the action pattern is suited to the animal's environment, or a functional motion rather than a haphazard random movement. The very special action pattern was hand to mouth. They had the monkey's hand placed in 11 different starting postures. They inserted an electrode into the precentral gyrus of the right hemisphere and applied a 100 microamp pulse at 200 Hz for 500 milliseconds. That is, for half a second. The electrode remained inserted into the very same region for the entire experiment. So each time, no matter where the starting position, the monkey's hand came to its mouth. Just to note, all during this experiment, the monkey was awake, and as reward after each electrical stimulation, happily reached for, grasped, and ate a raisin with his experimental hand. The monkeys apparently were not bothered by the artificial movement of their arm. An advance on the first major breakthrough with the canine's cortex with Fritz and Hitzig in 1870. Maybe the motor cortex has a GPS home address for the mouth, or perhaps a built-in or learnt proprioceptive location for the hand to mouth. But what it can't be is muscle-driven. Think about this. The same neural network, which can only have one outcome, is stimulated each time. Yet there is varying muscle recruitment to achieve the exact same end result, independent of the starting position. The motor cortex is clearly not sending motor commands to muscles. It's just not possible for one neural network to have 11 different actions. So what is it? What does the motor cortex deliver to the motor system for action? What about proprioceptive signals? Proprioceptive feedback from the hand, arm, and mouth is exactly the same at the target location, no matter the position from the hand at the start. Bingo! There is a match. Maybe the motor cortex knows or predicts what the proprioceptive feedback will be when the hand reaches the mouth. In this scenario, based on the proprioceptive prediction, muscle recruitment can be performed via the stretch reflex at the spinal level. This proposal that the motor cortex produces proprioceptive predictions comes from the brain theory called active inference. We will be hearing a lot about active inference. It is coming. We will take a closer look at active inference in another video. Graziano had extended Penfield's homunculus from a mere body map to an action map, a functional or ethological action map. It had been the belief that a prolonged stimulation would lead to widespread stimulation of the cortex, causing contamination and co-contraction of muscles, producing random movement. As well as 500 millisecond duration impulses, Graziano and team tried 20 millisecond brief duration stimulus, which Ferrier had found only produced muscle twitches and not functional action patterns. They also used 1000 millisecond impulses, that is 1 full second of stimulation, to produce the same functional patterns as the 500 millisecond stimulus.